Good afternoon. My name is Kim Cook. I'm a National Program Leader for Food Safety at the USDA Agricultural Research Service. I've been with ARS for over 16 years as a research scientist, a research leader, and a national program leader. My focus has been mostly on pre-harvest food safety and antimicrobial resistance. I wanted to start by thanking the committee for the invitation to talk about pre-harvest strategies to manage Salmonella. I did change the word control to manage, since I think management strategies, especially in pre-harvest production, have a greater opportunity for success at reducing Salmonella in post-harvest than do control strategies. Next slide. I wanted to start by saying a little about the ARS program structure since I think it's relevant. We have 4 program areas: crop production and protection, animal production and protection, natural resources and sustainable systems, and nutrition, food safety, and quality, where I work. And we work across 5 geographical regions that you can see in the map up on the right-hand corner. I mention this because our work is inherently farmer and stakeholder focused. So these regions and their locations focus on regionally relevant topics: forage in the plains area, produce in the west, poultry in the southeast, for example. These national programs and program areas interact to enable a multidisciplinary systems-based approach to address agricultural issues of a high national priority. And it is in fact critical to have multidisciplinary work in pre-harvest. It is incredibly diverse. The climate, topography, each of the commodities and its management differ within and across the commodity. Next slide. So I'm happy to address this topic. In fact, when asked, what is the one thing that my food safety stakeholders want most, my answer is always to solve Salmonella. My focus, therefore, is always on what does that look like and where can we make the most new impact? Current thinking about food safety approaches and where we can see the most new significant impact has turned much attention to pre-harvest food animal production. But the fact is that food safety is rapidly evolving across the continuum. Food safety is going beyond just surveillance and prevalence and beyond even genomics to thinking about metagenomics, artificial learning, and machine learning approaches. In food safety, the remaining hurdles, emerging pathogens, reducing illness, are complex, as this committee is well aware. In pre-harvest, it is even more complex, as I will discuss. So integrated approaches are needed for addressing food production through consumption. Pre-harvest and post-harvest are not separated, but considered an integrated production system of safe and quality food. Interventions that happen in pre-harvest affect the other segments in the food production system. So my focus is on holistic food systems thinking, which is needed to make new strides in salmonellosis, and that is what I'll talk about today. Next slide. There are opportunities to apply thinking, new thinking and novel approaches to manage Salmonella in pre-harvest. Entry point opportunities to stop it before it reaches processing facilities or any downstream processes. So new technologies, some I'll talk about, which can be leveraged. Chemical, physical, and microbial data are increasingly available and in collaborative environment can be leveraged through data assessment, predictive analysis, and targeted approaches that are increasingly the norm. However, there are also difficulties. Pre-harvest is not regulated, it's not uniform. We require good relationships with farmers that are forward-thinking and will assist with the burden of the science, which takes time and resources that are important commodities for farmers. There's incredible complexity. The graphic to the side shows the gridded sampling layout needed for statistically relevant sampling for foodborne pathogens across the beef production environment at the Meat Animal Research Center in Clay Center, Nebraska, a location with 34,000 acres of irrigated land and 8,000 heads of cattle. Approaches, therefore, have to be tailored for every farm due to differences in geography, integrator requirements, state and local laws, of which all can affect the outcomes. There's certainly no one size fits all in pre-harvest, but there are opportunities to identify mitigating factors through landscape analysis, predictive modeling, and risk analysis, and to identify and apply targeted, reasonable, and reproducible interventions that positively affect post-harvest outcomes. Next slide. So this is the first time that I've had a slide available for this, but I often ask the audience to imagine a salmonella that enters the food continuum either through a breeder's, the hatchery, or feed ingredients. The bacterium, whether in the bird's gut or external on feathers, in the litter, in the truck, facility, or surfaces, experiences an incredible diversity of conditions: moisture, temperature, pH, bacterial composition and competition, animal interactions through stress and diet changes. I'm just scrolling through these for the sake of time, but suffice it to say that it is a formidable opponent, and that makes it through all of these systems to make a consumer sick. So we have to be cognizant of the fact that what we are looking for in pre-harvest may be significantly different than what we focus on in post-harvest surveillance environments. Again, there are targeted opportunities in pre-harvest, including identifying pre-harvest indicators that predict post-harvest outcomes. And those may not even be the pathogen, they may be different. But we need to identify critical control points that work across commodities and correlate with those post-harvest outcomes. We need to identify tools that address emerging pathogens and identify interventions that are targeted, reasonable, and reproducible. Next slide. And so just to throw another what I think is a pre-harvest opportunity out there, wildlife, insects, and rodents are possibly the least understood pathway of transmission of pathogens and associated antimicrobial resistance through food systems. We can put in all the biosecurity measures we want, but nature will find a way. And this is an area ripe for focus, and I think more people are focusing on it. Next slide. So systems approaches are critical. They're complex, costly, and require multidisciplinary technology-focused activities. We need groups of out-of-the-box thinkers with knowledge across food systems, and the work in pre-harvest needs to be part of multi-hurdle approaches across the food continuum. I'll mention a few specific opportunities like the risk mapping and landscape analysis, including predictive microbiology and risk science, to identify gaps and opportunities in interventions, omics, gut function in the animal, and application of machine learning and remote sensing. I won't touch on innovative detection technologies, since I know you'll hear from others, including Dr. Dana Harhay from the ARS Meat Animal Research Center. However, I do want to add that that it's important that these innovative technologies take into account pre-harvest food safety and work across the food chain. Next slide. So it's important in pre-harvest to talk about data analytics and risk analysis. How do we get meaning from the volumes of data? Can we use predictive models to identify commodity-specific or even better cross-commodity opportunities for interventions? Risk models and risk science are needed to determine the impact on human health outcomes. Pre-harvest interventions will not be successful if they are not targeted. Next slide. Although mentioned more often in post-harvest, pre-harvest work also focuses on omics, AI, and machine learning. New approaches are moving from a focus on a single gene or class of genes like virulence factors to explain phenotype, to using models to understand not just if there is a gene or organism, but how we can predict phenotype and metabolic pathways that might serve as targets for interventions at different points in food systems. I often mention work by Dr. Sean Pearson at the ARS National Animal Disease Center in Ames, Iowa, that show that the outbreak strain of Salmonella redding that is plaguing the turkey industry actually has a reduced genome. So it did not gain new genes that made it more persistent, The emergent strain, it was not more competitive in pre-harvest environments when compared with a pre-outbreak strain, but might have been selected due to lower sensitivity to things like bacteriocins produced in competitive microbial environments. So we cannot just rely on the genetics, since survival factors for pathogens are based on fitness factors that are spatially and temporally specific. This also shows the value of animal models, which I'm a big believer. There are also opportunities to identify emerging pathogens, issues with pathogens in pre-harvest with things like vision-based monitoring tools, machine learning to predict negative outcomes, and notification systems for farmers. These may be more far-fetched since it will be difficult to identify pre-harvest monitoring factors that can predict post-harvest outcomes. And these approaches will almost certainly have to be commodity and even location specific, but they're definitely worth a mention. Next slide. So ARS recently finished our project planning for the next 5 years, and I was actually surprised at the large number of pre-harvest food safety projects that are focusing on things like vaccines that target swine and turkey, salmonella in swine and turkey, and they're able to differentiate infected and vaccinated animals, so-called DIVA vaccines. There's a lot of work on host pathogen interactions, evaluation of the gut biome and the host biome interactions to enhance gut function and exclude foodborne pathogens. Work to breed on breeding to enhance immunity to foodborne pathogens, and immune therapy and enhancement to exclude pathogens. Next slide. Finally, in the words of Dr. Sean Pearson in Ames, Iowa, who I will quote in full on the next slide, optimizing salmonella control strategies at each production and processing stage will offer the greatest possibility of successfully improving food safety and public health while diminishing the need for product recalls. Incorporating pre-harvest to post-harvest controls, iterative and multi-hurdle approaches will result in a more holistic opportunity to address salmonella challenges across the food chain. The pre-harvest challenges are formidable, but new technologies offer opportunities to do the hard work of using multidisciplinary groups to identify the public health risk, those that influence post-harvest outcomes to the greatest extent, to conduct statistically valid, technology-intensive studies across the pre-harvest environment and identify the critical control points and targets for interventions. Next slide. So finally, I wanted to provide the full quote from Dr. Pearson's upcoming review of salmonella in pork. And I wanted to thank you for the opportunity to speak and for your time, and I look forward to our discussions. Thank you.